already seen 4 stages of the meiosis 1 of prophase 1. Now we are going to see the diplotin and diagenesis, the last 2 phases of the meiosis 1 of prophase 1. So the beginning of the diplotin is recognized by the dissolution of the synaptic complex and the tendency of the recombined homologous chromosomes of bivalents to separate from each other except at the sites of crossovers. Now these X structures are called chiasmata. The oocytes of some vertebrates diplotin can last for months or years. Now the final stage of meiotic 1 prophase 1 is diagenesis. This is marked by terminalization of chiasmata. Now during this, mitotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diagenesis, the nucleus disappears and the nuclear envelope also breaks down. Now diagenesis refers to transition to metaphase. Now this was it. Now in this side you see recombination between homologous chromosomes is completed by the end of packaging. Recombination between homologous chromosomes is completed by the end of packaging, leaving the chromosomes linked at the sites of crossing over, and crossing over takes place during packaging between the non-sister chromatids of homologous chromosomes, and recombination is the another term. So this was all things, 2 things happening in the packaging. Remember, the crossing over between the non-sister chromatids of homologous chromosomes and then the recombination between homologous chromosomes is completed. Leaving, the third thing is also included, leaving the chromosomes linked at the sites of crossing over. This was packaging. And diplotin is dissolution of synaptic complex and the tendency of the recombinant homologous chromosomes or bivalents to separate from each other except at the sites of crossovers. So chiasmata formation during the diplotin, and diagenesis is the terminalization of chiasmata. How this was things. So this was the meiosis 1 of prophase 1. That includes leptotin, that is compaction of the chromosomes, the zygote synaptonemal complex formation, synapsis process includes bivalent and tetrad, and packaging includes crossing over, that is recombination, leaving the chromosomes linked at the sites of chromosome crossing over, and fourth one is diplotin contain chiasmata, shape structures, and diagenesis influence terminalization of chiasmata. Okay.